Electron micrograph pictures of some specializations or modifications of the cell membrane. Examples of apical surface specializations, microvilli, are finger-like projections from the free surface of the cell to increase the surface area. Stereocilia are not cilia but long microvilli, also increase the surface area of the cell. An example of lateral surface specialization, junctional complex, is present between two adjacent cells to fix them together. An example of basal surface specialization, hemidesmosome, is half of the desmosome and helps to fix the cell to its basement membrane and underlying connective tissue. Modifications of the apical surface, microvilli, finger-like projections on the free surface of the cell. Notice the presence of junctional complex between adjacent cells. Stereocilia, not cilia but long microvilli, present in the male reproductive system. Notice the presence of spermatozoa in the same photo. Microvilli by electron microscope appear as finger-like processes projecting from the upper surface of the cell membrane. There are two adjacent cells covered with microvilli connected together by junctional complex formed from zonula occludens, zonula adherens, and desmosome. Stereocilia are not cilia but long microvilli present in male reproductive system like the epididymal tube to increase the surface area of the cell. Electron micrograph photo of epididymal tube showing stereocilia as long cylinders extending from the free surface. Notice the presence also of spermatozoa in the lumen of the epididymal tube. Modification of the lateral cell membrane. Junctional complexes appear under light microscope as short dense lines. By electron microscope, each is formed of tight junction zonula occludens, adherent junction zonula adherens, and macula adherens or desmosome, formed of two dense cytoplasmic plaques to which are attached multiple tonofilaments. Notice the presence of microvilli on the free surface of the cell, together with cell coat or glycocalyx. Secretory granules appear as electron-dense granules near the top of the cell to be discharged, while ribosomes appear as small dense granules in the basal part of the cell for the synthesis of the proteins and the secretory products. Modification of the basal cell membrane. Hemidesmosome consists of one half of a desmosome formed of one cytoplasmic plaque with abundant tonofilaments or keratin filaments embedded in it. Hemidesmosome fixes cells to their basement membrane and to the underlying reticular fibers. Many ribosomes are also seen in the photograph. Pseudopodia are outward projections from the cell membrane, numerous in phagocytic cells, helping them in the process of phagocytosis. Electron micrograph photo of a phagocytic cell showing the presence of two nuclei, multiple pseudopodia, and specific granules.